Hello, my dear students. Welcome you all to Sri Jayendra Rank Zone classes. I'm your mentor and your friend for the subject biology, and I will be taking the zoology part, that is animal science. My dear students, in the previous lecture, we have started body fluids and circulation. We discussed about the arteries, veins, their histological features. We discussed about heart and different chambers. Today we are going to start with the capillaries. That was the last part which was left to cover in the last lecture. Capillaries. It is a very fine blood vessel where the wall is made up of single layer of endothelium of tessellated cells. Tessellated cells means the cells or the epithelium is basically is an arrangement of cells having some pores. They have some pores. Okay. So this tessellated epithelium helps to control the amount of tissue fluid. Helps to control the amount of tissue fluid. Right. Okay. So it's a very fine blood vessel where the wall is made up of single layer of endothelium of tessellated cells. Fine intercellular Fine intercellular cleft occurring between adjacent endothelial cells. Okay, the endothelial cells, they are arranged with some intracellular clefts. Okay, that is why these intracellular clefts, presence of these intracellular spaces of clefts, make the epithelium very specialized, and that is why it is known as what? Tessellated epithelium. Basement membrane lies on the outside. Okay, basement membrane is, it lies on the outside part. Okay, blood capillaries are formed by arterioles. Blood capillaries, they are formed by arterioles. They join to produce venules. Okay, so blood capillaries basically they are formed by arterioles, and the arterioles they join venules to produce these capillaries. The lumen of blood capillary, the lumen means the space through where which the blood flows, or the blood capillary, is so fine that red blood corpuscles can pass through it in a single file. Mean it is having a space equivalent to one single cell passage. Only one cell at a time can pass through it. The blood capillaries, they are formed by arterioles. They join venules. So arterioles and venules, they join to form blood capillaries. Okay. The lumen of blood capillaries is so fine that red blood corpuscles can pass through it in single file. White blood cells can come out of them. White blood cells can come out of them through the process of diapedesis. Diapedesis means marginal movement. The white blood corpuscles, they can come out through these capillaries through the process of diapedesis, that is marginal movement, because of their extremely thin walls. As blood capillaries, they have extremely thin walls, the WBCs of white blood cells can easily cross through the membrane. Blood capillaries take part in exchange of materials. It take part in exchange of materials between blood and tissue fluid. Okay, so between blood and tissue fluid, whenever is required, the exchange of gases, exchange of nutrients takes place. Okay, so capillaries, they help basically to make this exchange happen. In lungs, they pick up oxygen and give out CO2 through diffusion. Because of these capillaries, the alveoli vessels or the alveolar part is highly rich, to exchange the gases, especially the oxygen and CO2 gas. All the blood capillaries are not functional at all time. All the blood capillaries, they are not functional at all times. Some of them work only at the time of intense activity. Some of them, they work only at a time of intense activity. Okay, whenever is required, some of them they work. Okay, their working is controlled by precapillary sphincters. Their working is basically controlled by pre-capillary sphincters present in the area of origin. Okay, so these capillaries where they originate, they have some sphincters, some guards, some valves. These pre-capillary sphincters, they help to control or regulate the functioning of these capillaries. Now moving to the next slide, arteriovenous anastomosis. Anastomosis means junction. Okay, it is a direct vascular connection between arteriole and venule. Arterioles and venules, they come up to make a direct vascular connection, bypassing the capillary bed. When they are not making capillaries, they are directly coming in contact with each other. The connection occurs in certain exposed parts like fingertips, nose, pinna, eyelids, lips, tongue. I mean, all the extremities of body, they basically have this arteriovenous anastomosis. As the capillary bed is bypassed, they are not going to frame a distinct structure like capillaries. They are just merging with each other directly. Okay, it is meant for controlling blood supply. It is meant for controlling blood supply and the temperature of exposed parts. Very important. These arteriovenous anastomosis, they basically help to control the temperature of these extremities. As you know, whenever it's extreme cold or extreme hot, okay, the fingertips, I mean, they all, the nerve endings, they lies here. So it is very important to control the body temperature, and that is why arteriovenous anastomosis is there, because capillaries could take more time to maintain the functions. Okay, so it is meant for controlling blood supply and temperature of the exposed parts. Vascular plexus, anastomosis of blood vessels like arteries in certain regions to provide extra blood. Okay, to provide extra blood, like cutaneous plexus, papillary plexus, and nasal plexus. Vascular plexus, what is vascular plexus? It is the anastomosis of blood vessels. I mean, when many blood vessels, they come together, okay, many arteries, they come together in a particular area to provide excessive blood, extra blood according to the metabolic requirement of that area. Okay, especially the cutaneous plexus, okay, papillary plexus, and nasal plexus. All the areas where the membranes are thin, or where we can say the temperature can directly influence that part, the blood supply should be rich out there to maintain the body temperature. So vascular plexus is clear to all, I think, and arteriovenous anastomosis is clear to everyone. Now, efficiency of closed respiratory system. Okay, why we always prefer a closed respiratory system over a open respiratory system? It can regulate blood flow into an organ. So this is closed circulatory system. Efficiency of closed circulatory system, it can regulate blood flow into an organ. Okay, blood flows rapidly in blood vessels than in open spaces. The first thing, it can regulate blood flows rapidly in blood vessels than in open spaces. There is quick supply and removal of materials. There is very quick supply, very fast supply, according to the high metabolic demand. Okay, so to meet out high metabolic demand, to eliminate major metabolic waste, we requires all these vessels. So there is quick supply and removal of materials. Blood flows to all parts of the body with equal efficiency and speed. Very important. Okay, blood flow to all parts of the body with equal efficiency and speed because of having this closed circulatory system. So because of closed circulatory system, the blood flow it can be distributed uniformly to all the, not uniformly according to the requirement to, you know, all the body parts. It travels with equal efficiency and speed. There is a quick supply and removal of materials. Blood flows rapidly in the blood vessels and in open spaces. It can regulate the blood flow into an organ. Okay, blood. Now we will talk about blood. Blood is also known as the fluid connective tissue, fluid connective tissue, or we can say the false connective tissue. So blood is also known as the fluid connective tissue or the false connective tissue. It is complex mobile fluid. It is a complex mobile fluid, mean it is a fluid, and it is a complex fluid which is colloidal in nature, which is colloidal in nature. Okay. It is a connective tissue which is colloidal in nature, complex and mobile, of reddish color, in which the fluid matrix is not synthesized by the contained cells, in which matrix is not synthesized by the cellular component of blood. That is why it is known as what? False connective tissue or fluid connective tissue, as there is no matrix which is synthesized by the cells. An adult human has around 5 to 5.5 liters of blood. pH of blood is 7.4, slightly alkaline. Blood consists of two parts, plasma and blood corpuscles. Blood corpuscles are known as what? Formed elements. Okay, formed elements and plasma. Plasma is a slight alkaline, non-living intercellular substance which constitutes about 60% part of the blood. It is pale, yellow, but transparent and clear. Okay, plasma, it is slightly alkaline, non-living intercellular substance which constitutes about 60% part of the blood. It is pale, yellow, but transparent and clear fluid. It is composed of 91 to 92% of water. It is mainly made up of 91 to 92% of water, 7% of proteins, 0.9% inorganic substances, 0.1% of glucose, and traces of other constituents like amino acids, fatty acids, like amino acids, fatty acids, fat droplets, cholesterol, anticoagulants, hormones, excretory products, vitamins. So it mainly composed of, plasma is mainly made up of 91 to 92% of water, 7 to 7% are proteins, 0.9% are inorganic substances, 0.1% of glucose, and traces of other constituents like amino acids, cholesterol, fat drops, fatty acid, anticoagulants, hormones, excretory products, and vitamins. The main categories of protein are albumins, fibrinogen, and globulins. Albumin is maximally present in plasma. Okay, among all the proteins, albumin is present around 3 to 3.5% out of 7% of constituency. Okay, albumins are present 3 to 3.5%. It maintains the colloidal blood osmotic pressure. It also carry calcium and some fatty acid like alpha globulin, beta globulin, carry fat soluble vitamins, cholesterol and ions, and other globulin are prothrombin, thromboplastin, and antihemophilic factors. Fibrinogen takes part in blood coagulation by forming fibrin. So albumin maintains the blood colloidal osmotic pressure. It also carry the you know calcium ions and some other fatty acids. It also carries with itself calcium ions and some other fatty acid. Alpha globulin, beta globulin, they help to carry fat soluble vitamins, cholesterol and other ions. Okay, other globulins are prothrombin, thromboplastin, and antihemophilic factors. Fibrinogen takes part in the blood coagulation by forming of fibrin. Mineral salts like chlorides, bicarbonates, sulfates, phosphates of sodium, potassium, calcium, iron, and magnesium constitute about 0.9% of the plasma. These all are inorganic salts which are present 0.9% of plasma, like salts of chlorides, bicarbonates, sulfates, phosphates, ठीक है ना? Especially of sodium, potassium, and calcium, iron, and magnesium constitute about 0.9% of plasma. Buffer of the blood is sodium bicarbonate. Very important. Buffer of the blood is sodium bicarbonate. Sodium bicarbonate. So this is about plasma and its constituents. Okay, its composition, functions of blood plasma. It helps in the transportation of the nutrients, retention of fluid in the blood. It helps to maintain the amount of volume. Helps to maintain the volume of blood, transportation of nutrients and metabolic waste. It helps to maintain the blood pH, very importantly, because it is having buffers like sodium bicarbonate. It helps to maintain the body immunity, reduces lymphocytes. It helps in the prevention of blood loss by making clot. Okay. It helps in the conduction of heat to skin for dissipation. To maintain body temperature. It helps in the uniform distribution of heat all over the body. So what are the functions of blood plasma? It helps in the transportation of nutrients and metabolic waste. It helps in the retention of fluid in blood, and it helps to maintain the volume of the blood. It helps to maintain the pH of the blood because it contains buffers like sodium bicarbonate, which maintains the amount of, you know, uh, or regulate the blood pH. Then it helps to maintain the body immunity because it carries lymphocytes with it, prevention of blood loss by making clot, conduction of heat to skin for dissipation, to maintain the body temperature, and it also helps in the uniform distribution of heat all over the body. Okay. Blood glucose level is about 80 to 100 milligram per 100 ml of blood 12 hours after a normal meal. So the blood glucose level which maintains as a loading dose after 12 hours having after a normal meal is 80 to 100 milligram per 100 ml, okay, or 82 milligram per deciliter. The blood glucose level exceeds 180 milligram per 100 ml if it starts appearing then it starts appearing in urine. So the threshold maximum Tmax. Threshold maximum is the value of maximum amount of blood glucose or any other substance which can be retained by nephrons. Okay, which can be retained by nephrons, especially the proximal convoluted tubule. Okay, so the maximum amount of substance or a reabsorbate which can be held by the PCT. Okay, is the maximum threshold or the threshold maximum Tmax. Blood, blood glucose, the normal range is 80 to 100 milligram per deciliter. Person is said to be diabetic if it is having more than 130 or 140 milligram per deciliter of blood sugar level. But if a person urine is analyzed or sent as a sample for the evaluation of sugar, then the urine or the appearance of glucose in urine doesn't appear before it in a range of threshold maximum. The moment it crosses the range of threshold maximum, then only the sugar or the glucose appears in the urine. The appearance of glucose in urine is known as what? Glycosuria. Okay, if it is less, it causes hypoglycemia, and if it is higher, then it causes hyperglycemia. This is in blood. If the levels of sugar level in blood is less than 80, then it is hypoglycemia 80 or 100, and if it is more, then it is known as what? Hyperglycemia. Blood cholesterol levels, the normal amount is 80 to 180 milligram per deciliter of blood plasma. Increased blood cholesterol may lead to its deposition in the internal wall of blood vessels like arteries and veins, which causes high blood pressure and heart problems. Means arteriosclerosis. Okay, so the normal blood sugar level is 80 to 100 per deciliter, blood cholesterol 80 to 180 per deciliter. Increased amount of blood cholesterol can raise direct or indirect bile salts and bile pigments. Okay, because bile salts, they are basically associated with the absorption of direct triglycerides. Okay, so direct or indirect bile salts, they appear, or their range, their amount changes in the blood because of change in the fat metabolism. Formed elements mean, when we talk about the packed cell volume, around 45% of packed cell volume, and the plasma around 55%. So formed elements, they are the cells which are present in the blood, like erythrocytes, also known as red blood corpuscles or RBCs. A normal adult man and woman have 5 to 4.5 million RBCs per cubic millimeter of blood respectively. This range can be, it is 5.5. In case of males or boys, it is around 4 to 4.5 million per cubic mm, and in case of girls, women, it is around 3 to 3.5 million per cubic mm. Less amount of hemoglobin leads to anemia. Very important. Less amount of hemoglobin leads to anemia, which may be caused by the loss of blood or destruction of RBC. Less amount of hemoglobin leads to anemia, which may be caused by loss of blood or destruction of red blood corpuscles. Abnormal rise in RBC count is called polycythemia. Polycythemia. Decrease in number of RBC is called erythrocytopenia, which causes shortage in the blood and tissues. They are RBCs are biconcave, disc-shaped, enucleated, reddish-colored cells of 7 to 8 micrometers in diameter and 1 to 2 micrometers thick. Red color is due to the presence of hemoglobin. Hemoglobin is a conjugate protein which is made up of a protein called globin and a non-proteinaceous part, that is the prosthetic group. Okay, heme. Heme, hemoglobin. Okay, heme is a iron conjugate prosthetic group. Okay, iron porphyrin ring. Okay, hemoglobin is a conjugate protein which is made up of protein called globin and a non-proteinaceous part. Okay, which is known as what? Heme, and together they are known as what? Hemoglobin. Hemoglobin is oxygen carrying pigment. Around 100 ml of blood of normal male contains 15 gram of hemoglobin, and of normal woman 13 gram of hemoglobin. Erythropoiesis is the process of red blood cells, okay, are produced. In human adults, this occurs only within the bone marrow, while in before birth or infants, liver, kidney, yolk sac, all are erythropoietic organs. So it is known as what? Erythropoietic organ. The lifespan of human RBC is around 120 days. For frog, it is 100 days. The worn out RBCs are destroyed in the spleen and liver. The spleen and liver, there represents the reticuloendothelial system, and they are graveyard or RBCs. Okay. The iron is returned into the red bone marrow for reuse in the synthesis of fresh hemoglobin. Their pigment is degraded to yellowish pigment bilirubin, which is excreted in the bile. Okay. This is the composition of blood. Whole blood can be divided into plasma formed elements. Plasma mainly contains amino acids, nutrients, proteins, albumin, globulins, and fibrinogen. Plasma also contains gases, diffusible gases, electrolytes, and nitrogenous waste. Formed elements: leukocytes, WBCs, erythrocytes, RBCs, and platelets, megakaryocytes or thrombocytes. Leukocytes, granulocytes, and agranulocytes. Granulocytes, neutrophils, eosinophils, basophils. And agranulocytes without granules, lymphocytes, and monocytes. Granulocytes are with granules. So WBCs or leukocytes are further classified into granulocytes and agranulocytes. Granulocytes are with granules, neutrophils, basophils, and eosinophils. And agranulocytes, they don't have granules, lymphocytes and monocytes. It also contains platelets. It also contains erythrocytes. So this is the classification of whole blood. In the next lecture, in the coming lecture, we will be discussing about distinctly about RBCs, WBCs, platelets. Okay? So till then, bye bye everyone. Stay safe, stay healthy. Bye bye.